Hereby, I open this academic ceremony in which Alicia Todaro will defend the academic thesis, Genetic Mechanisms of Inherited Bleeding Disorders as a Basis for Personalized Medicine Approaches. May I invite you to present a summary of your study and your conclusions. And take your time.
Thank you very much. Dear Prorector, members of the Assessment Committee and the oppositions, supervisors, family, friends, and colleagues here on site and online, thank you for being here, and welcome to my PhD defense. In the next 15 minutes, I will present an overview of my PhD research entitled Genetic Mechanisms of Inherited Bleeding Disorders as a Basis for Personalized Medicine Approaches. The basis of my research begins with the hemostasis, which is the process that stops bleeding. Normally, the blood flows into the vessels, but when we damage our skin, for instance, we all experience that we lose blood, so there is a blood loss. But after a couple of minutes, usually we stop bleeding, and eventually the vessel is repaired. This process of stopping the blood loss is because there is a formation of a clot. And now we'll go a bit deeper in what is exactly the clot formation. So the hemostasis, so the clot formation, is actually divided in primary and secondary hemostasis. In the primary hemostasis, platelets sit on the injured vessel. And for this process, von Willebrand factor, or VWF, is really important. The secondary hemostasis, or coagulation cascade, has as a main goal the formation of fibrin. So fibrin, which is represented here as these fibers, stabilizes the platelet plug, therefore to help in the clot formation. Fibrin is formed by the coagulation cascade, which is a very complicated process, and here is shown in a simplified version. With the formation of thrombin, which is the enzyme that converts fibrinogen into fibrin. Of course, the hemostasis process and also the coagulation cascade needs to be stopped at some point. And there are many inhibitors of the coagulation cascade, and one of them is called TFPI, or tissue factor pathway inhibitor. And TFPI, factor 5, and factor 8 are important for this presentation and for my research. That's why they are underlined here. And in particular, in my research, I focused on mutations impairing von Willebrand factor and factor 5, and these mutations lead to bleeding disorders. In chapter 2 of my thesis, we studied a von Willebrand factor patient with a severe bleeding disorder due to von Willebrand factor abnormalities. But first, what is von Willebrand factor and what is von Willebrand disease? Von Willebrand factor is very important in the primary hemostasis because it activates and helps in the adhesion and aggregation of platelets at the injured vessel. But it also maintains coagulation factor 8 in the circulation. Therefore, there is a binding between von Willebrand factor and factor 8. However, if mutations impair von Willebrand factor, this results in VWD, or von Willebrand disease, which is a common bleeding disorder. We investigated a female patient with a severe bleeding disease with excessive bleeding and 2 miscarriages. From the laboratory tests, her von Willebrand parameters were very low, or anyways lower than the reference range. Therefore, pointing at von Willebrand factor as one of the main issues for her bleeding disease. Therefore, we investigated the genetics of this patient, and in her von Willebrand factor allele 1, we found a novel mutation which led to the production of von Willebrand factor, but this von Willebrand factor is dysfunctional. And in particular, this dysfunctional von Willebrand factor impairs the binding between von Willebrand factor and factor 8. And this led to von Willebrand disease of type 2N. In the other allele, we have strong suggestions from our messenger RNA analysis that there is a mutation there. And we don't know yet which one it is, but we know the result of this, which is no von Willebrand factor protein expression, which accounts for less von Willebrand factor protein, therefore a von Willebrand disease of type 1. In chapter 3, we investigated a patient with a lack of coagulation factor 5 and tried to correct the underlying genetic defect. But first, what is factor 5 and what is factor 5 deficiency? Factor 5 is the essential cofactor of activated factor 10 in the coagulation cascade in order to form thrombin, which is the enzyme that converts fibrinogen into fibrin. If mutation strikes factor 5, this leads to factor 5 deficiency, which is a rare bleeding disorder. In particular, we studied a patient with almost no coagulation factor 5 and a moderate bleeding tendency. We studied in particular the genetics of this patient, and we found that on both allele of this patient's factor 5, there is a stop mutation. A stop mutation is a very strong signal for the impairment of protein expression. However, we knew from literature that there are some compounds that are able to bypass these stop mutations, therefore restoring a full-length protein. And we were wondering if this was also happening for factor 5. Therefore, we applied a possible correction approach. Cells expressing the mutated factor 5 were treated with compounds that are able to bypass the stop mutations. And we used some of them at different concentrations. And what we observed was a progressive restoring of factor 5 expression, but also function. We also had the chance to work directly with cells from the patient. So we collected cells from a control and the patient that were able to express factor 5. And as you can appreciate here, the cells from a control can normally express factor 5 here detected in green, while patient cells only had the nuclei visible. We treated these cells with the stop bypassing compounds, and after treatment, we observed that the cells from patients which before were not expressing factor 5 now can express factor 5 again. In chapter 4, we studied a variant of factor 5 called factor 5 short, which is associated with a bleeding tendency. Factor 5 short is an isoform of factor 5, meaning that it's a similar form but a little bit different. And to be able to explain the difference, I put the small scheme on top left because the genetic information usually goes from DNA through the messenger RNA to eventually form protein. So now to explain the difference between factor 5 short and factor 5, I will talk about the messenger RNA. In particular, the pre-messenger RNA. Pre-messenger of RNA, RNA of factor 5 can undergo to two different mature RNA. The first form, which is also the most abundant, in this form, after splicing, which is a process that removes what is not necessary to codify the protein, the entire exon 13 of factor 5 is retained, giving a full-length factor 5 protein. However, if two splicing sites called donor and acceptor are active, the light blue part is cut out of the messenger RNA, therefore giving a shorter version of both messenger RNA and then protein, called factor 5 short. Factor 5 short has the characteristics of binding with high affinity TFPI, or tissue factor pathway inhibitor, which is an inhibitor of coagulation. Therefore, this complex confers to factor 5 short an anticoagulant property. To date, there are 3 mutations that upregulate factor V short. So when factor V X Texas, Amsterdam, and Atlanta mutations are present, there is an overexpression of factor V short. So more factor V short, therefore more TFPI, and this results in a bleeding tendency with excessive bleeding after trauma. Therefore, we try to downregulate, so to decrease factor V short, and here we applied first one strategy based on morpholino antisense oligonucleotide, or MAO. Therefore, we tried to design antisense oligonucleotide donor and acceptor exactly on the spliced site to block the formation of factor V short messenger RNA, with the goal to down-regulate factor V short. And we tested this hypothesis. So liver cell line already expressing factor V and factor V short were treated with specific MAO molecules at different concentrations. And after treatment, we observed a progressive downregulation of factor V short expression when using donor MAO and acceptor MAO and a combination of them here in the mix, but no downregulation of factor V short expression when using a control MAO. In chapter 5, we tried a different strategy, always with the aim to downregulate factor V short expression, but this time also to downregulate factor V short related bleeding disorders. And this strategy, this time, involves the mature messenger RNA of factor V short. We used siRNA, or short interfering RNA, which targets directly and specifically factor V short messenger RNA, leading to messenger RNA degradation, therefore less factor V short expression. And we tested this hypothesis. Cells that were expressing factor V short variants were cultured and treated with specific siRNase molecules. And after treatment, we observed a progressive downregulation with an increase of siRNA concentration for all the variants of factor V short. This leads to the conclusions of my presentations. So regarding von Willebrand factor, in chapter 2, we identified and characterized a novel mutation, R760S, in the von Willebrand gene of a patient, and this new mutation impairs the binding between von Willebrand factor and factor VIII. Regarding factor V deficiency, in chapter 3, we studied a patient with almost no factor V, and we corrected the stop mutation by using compounds that were able to restore the synthesis and also the function of factor V in both cells and also in vitro model and also cells from patients. For factor V short in chapter 4, we were able to downregulate factor V short expression in a liver cell line model by using morpholino antisense oligonucleotides. And in chapter 5, we downregulated factor V short variants, so wild type and also the variants that led to a bleeding disorder, by using the different strategy based on siRNAs in cell line models. With this, I would like to thank you for your attention, and I give the word back to the prorector.
Thank you for your very clear presentation. The opposition will be opened by Professor De Wind, Professor of Molecular Cardiovascular Biology at this university.
Thank you, Mr. Prorector. Dear graduate, congratulations on this festive day, where you can celebrate your accomplishments of this very impressive thesis that was also very pleasantly written, and that shows the results of very hard work in the past years. Of course, I also salute and congratulate Dr. Castoldi and Professor Tillman for making this journey possible. And I certainly also want to stand still for just a few seconds and congratulate your friends, your family and loved ones for their help and support in the past years. Quindi soprattutto le mie congratulazioni ai tuoi genitori. Avete una bravissima figlia che ha raggiunto un ottimo lavoro e spero che abbiate un bellissimo giorno oggi di festa. Okay, graduate, I wasn't invited only just to give you compliments. We also have some work to do. And for this, I would like to exchange a bit our thoughts on the work presented in chapters 4 and 5. And in chapter 4, you show that you can achieve knockdown of an alternatively spliced short isoform, as you presented in the presentation. It was really beautiful, with a 20 nucleotide morpholino oligomer. And these DNA morpholinos contain a phosphorodiamidite groups instead of a phosphate group to stabilize the molecule, and they work by steric hindrance. They work by steric hindrance. There's an antisense oligo that works by steric hindrance. So my first question would be, why the choice of a morpholino oligo? Can you elaborate maybe?
Highly esteemed opponent, thank you for your kind words for me and for my family and for my thesis and for your question. Why morpholino? So morpholino, we chose morpholino because we also saw from the literature that it was working for the purpose to downregulate the undesired messenger RNA. We wanted to achieve. And we also knew from literature that it was very stable.
Yes.
And one of them is actually approved for Duchenne muscular dystrophy.
Yes, yes.
So
the name is, I think, let me see, there was, Golodirsen.
Golodirsen.
By Sarepta Therapeutics.
So because, of course, our final goal, hopefully one day, would be to treat patients, we thought to start already with, hopefully well-working molecule, but also safe, already showing safety in patients, and
Yes.
It is also not charged, so also the charge could impair the entrance in the cells and then the work. So
Yeah, maybe we have some time afterwards to talk about the patients, but I would want to go back to the molecular aspects of an antisense that is very stable, as you said, and it binds then, like this, binds to the messenger RNA that you don't want. But then what do we measure? Because there was figure 6, I think, I'm not so sure. No, figures 4, 5, page 87, 88, you develop a qPCR assay, and you actually demonstrate that you have less of your undesired short messenger. But what are we measuring now?
I see what you mean. So
You see what I mean?
yes. So morpholino molecules, they
Yeah.
bind on the pre-messenger RNA.
Okay.
So they compete with the spliceosome. So we then measure in the qPCR the actual factor V short messenger RNA. Because, of course, if you have morpholinos and then they work, they avoid that the spliceosome can use those
Attach.
splicing sites. Exactly.
Okay.
So they compete with the spliceosome. So what we measure is after splicing.
Okay.
So we go and measure the mature messenger RNA.
Perfect. Then we're on the same page, because if a qPCR would still contain a double-stranded RNA molecule, then you cannot generate a proper copy DNA, and your PCR assay will not.
Exactly,
So, okay.
but we
Beautiful.
measured after that. So
Yeah.
we were sure that what we were measuring was actually
yeah.
messenger RNA after all the splicing process.
Congratulations, promotion team. This is a very good candidate. So then maybe we can chat a little bit about the siRNA approach, which is quite different. siRNA. Now you really make use of the interference pathway, so you degrade the smaller siRNA. Which one would you prefer now? Looking back at all your work, which one do you think would be more translatable to patients? An antisense approach or an RNA degradation approach?
I think right now I would pursue siRNA strategy to downregulate factor V short, in this case, messenger RNA, because there are many studies on different siRNAs already in clinical trial. There is actually one also for coagulation, fitusiran is called, and it showed very promising data from clinical phase 3. And it is against the antithrombin. So it showed a very nice profile also in patient, and it was working. And some siRNAs, 5 siRNAs are approved by FDA or EMA for treatment in patients. So I think I would pursue siRNAs research right now with this purpose.
Yeah, I'm looking at the prorector. Do I have half a minute left? Half a minute, yeah. So could you make a step from your in vitro studies where you use micromolars of your siRNA towards humans? What would be the dosage that we would have to use in humans?
Sorry, I didn't get what would you?
So if you use 30 or 50 micromolars of your siRNA, and then we have to calculate what doses will I give to a patient, assuming that all these RNA molecules, they will always end up in the liver, so it's 100% in the right organ. Which dose are we thinking then?
So I'm not a medical doctor, but I was reading in literature that, for instance, for fitusiran, they give 80 milligrams of this and of siRNAs, which is usually given subcutaneous or intravenous. 80 milligrams they use once a month.
Okay.
So I guess around that, or maybe start even lower just to be sure for safety profile if it's a different siRNA.
Thank you for the interaction. Mr. Prorector, I give you back the word.
Thank you for your questions. The opposition will be continued by Professor Bekers, Professor of Clinical Chemistry in Maastricht University.
Dear candidate, I want also to start with something that's for me very different, difficult to say, but I will try it. Congratulazioni per aver completato tua tesi. E questo vale anche per il tuo colorato. And for your promoter, I will give my compliments in English. Besides the very interesting content of your thesis, I also want to wish you compliment with the design of your thesis, and especially the front cover is molto bello. But of course, I'm not here to give you only compliments. I also want to discuss some issues with you. And I want to imagine that I was a director of a pharmaceutical company, and I wish to develop molecular therapy for personalized medicine. What would you, as an expert in this field, advise me to do to bring this kind of therapies to the patient care?
Highly esteemed opponent, thank you very much for your kind words and for your question. So if I got it right, your question, you're asking whether from a pharmaceutical company point of view, you would
bring your what you have studied things, and how can we bring that to the patient care? What kind of steps we have to do? You could advise me when I was a director of a pharmaceutical company. I have a lot of money, so I ask you, how can we bring your study to the patient care?
To the patient. Okay, so what I did with the morpholino and siRNA, of course, is preclinical, and especially the siRNA is still preliminary report. So for the siRNA, from this point of view, for factor V, I would say more experiments, so that can connect the in vitro, so what I did, to more clinical outcome. So for instance, I would proceed with or an ex vivo, or for instance, iPSCs from a patient with, in this case, a patient with the overexpression of factor V short, to to see whether this also works with cells from patients, or if not available, I would also start from the iPSCs wild type, and then I would insert the mutation and then try to correct that. So I would say
starting with cells.
As first step, I would try with cells, and usually the further step is animal model. Usually, but for factor V is a bit complicated because, for instance, in mice have different factor V than humans, and because factor V and factor V short are also associated with TFPI in humans, while in mice this association is not detected.
You could choose other animals, probably?
I've been reading that some researchers are trying with zebrafish, although it's far from mammals. So for factor V it would be very difficult to find an animal model, at least for the correction. For safety, you could use a mouse or another animal model for pharmacokinetics or bioavailability or safety profile, and also for clearance for molecules. So for all this part, which is also important for a molecule because before it reaches human, you can use an animal model. For factor V correction is a bit tricky.
Mm-hmm. And if you go to a timeline, do you expect how many years such therapy will be used?
I hope as soon as possible. Maybe for siRNAs, I think it's difficult to estimate how many years, but probably less than starting from zero with a completely new molecule, I would say, because on siRNA safety and bioavailability and all these characteristics, there are a lot of data and a lot of clinical trials and studies. So probably with those on records, you could gain some years, I would say.
Correct. Can I ask one more question? Well, I'm a clinical chemist, and you're an expert also in factor V. And in our laboratory, we do hundreds of tests of factor V Leiden. You probably will know that. Do you think that is a useful test? And do you find that it has a place in diagnostics? Because there was a lot of discussion about factor V Leiden. We have to perform the test, or didn't we have performed the test? What's your opinion about that?
I'm not sure if I got the question right. So if we test, if it's factor V Leiden?
Yeah, we tested a lot in our laboratory, but there are some people who said that it's not necessary to test the factor V Leiden.
Okay, I think it depends. So if a person has already having symptoms, so factor V Leiden leads towards thrombosis. So if there is something related to that, I would test for factor V Leiden. And
all the patients?
All patients, I think it depends by the goal. For instance, if a person, the first thing that comes in my mind is, for instance, pill for women. Some of them it's known that they are procoagulant. So in that case, I would test for that, just to be sure. And so then the patient would be aware. Or other therapies, or for instance, also surgery. I think it would be important to test whether there are factor V Leiden or other procoagulant issues, I think.
Okay, well, I'm satisfied with your answers, and I give the word back to the prorector.
Thank you. The opposition will be continued by Professor Knosse, Professor of Pediatric Hematology, Erasmus University. Thank you for coming all the way from Rotterdam.
Thank you very much, Mr. Prorector. What an honor to be here today at the public defense of your doctoral thesis, dear candidate. Of course, before I go on, I want to congratulate you and your promotion team on the results of your endeavors, resulting in this very interesting thesis. My first acquaintance with your co-promoter, Dr. Elisabetta Castoldi, was directly after a wonderful session on Factor V at an ISTH conference some years ago. We stayed in contact, and therefore it is extra special to be here today and to applaud this thesis, in which 3 of the 4 chapters are on factor V developments. These developments are especially important as therapeutic options for severe factor V deficiency are limited, and I know that as a pediatric hematologist. Long-term treatment with plasma causes severe nephrological complications due to the immense protein load, and I've seen patients suffer. Before I go into chapter 3, I'd like to ask you a kind of a different question, and not in Italian. Please excuse my lack of Italian, my lack of knowledge of the Italian language. Do you know what alternative names are for severe factor V deficiency in a historical context?
Highly esteemed opponent, thank you for your kind words and for your question. Other names for factor V deficiency. Factor V deficiency is also known as parahemophilia, historically, and as Oren's disease.
Oh, very well done.
From the discoverer of factor V.
Yes. Did you know that he was also a fox farmer?
Fox farmer? No.
Something left to learn. Thank you very much, and I'm very impressed that you know this. He defended his thesis in 1946 on factor V deficiency. So the historical context is very important. You've really advanced the field. Now for my questions on chapter 3. You looked at a patient with detectable levels of natural read-through, factor V, which is very important, of course. And you looked at 5 agents in an in vitro and an in vivo setting. The results of these agents were quite impressive. The first agent you used, the G418, gave a 7-fold readout of factor V, the ELX02, 3.1-fold, and the PTC124, 10.8-fold, which is also very impressive. The next 2 agents were ineffective. In your discussion, in your results, you say that these effects were confirmed by a thrombin generation assay. And further on in your article, you suggest that this may be a dose-effect response. Looking at figure 3 on page 59, I am not very impressed by the thrombin generation assay results, and I do not see a dose-response effect. Could you please explain?
Okay, yes, figure 3. Okay, so yes, using indeed G418, ELX02, and 2,6-diaminopurine, we actually measured that factor V expression was restored and also the function. And in thrombin generation is, okay, you're not impressed with the results in thrombin generation. So you were expecting more in
now,
thrombin
especially
generation.
the PTC124. I'm just a simple pediatric hematologist, of course, but I know how thrombin generation assays look like. Should show a 10.8-fold increase of factor V effect. And looking at that assay in figure 3b, the thrombin generation hardly increases, doesn't it? Or is that not? And could you please explain? Yeah.
Yes, of course. The thrombin generation curve does not reach like a normal total functioning coagulation factor V. So in this thrombin generation, of course, in the thrombin generation is more physiological, like this experiment, compared to the graph above. So the graph above is in buffers, and there are pure proteins interacting, while in thrombin generation we used factor V deficient plasma. But of course, all the rest of the coagulation factors and inhibitors are present in that plasma. So it could be that they also play a role in this thrombin generation.
So the fact that it's more physiological is what you're actually telling me, that's what you're showing in the assay. Yeah.
Yes, yes.
Okay.
While the graph above, the one in gray, there are pure proteins interacting there.
Wonderful. Thank you. The next question, you're talking about a cytotoxic response that you see in the agents. And I was wondering, do you know something more about the mechanism behind this cytotoxicity? Especially as you tell us in your discussion at the end of your thesis that 2 of these agents are currently being used in rare diseases, therapeutically, in Duchenne's disease and in Alport disease. What do these results tell us on this cytotoxicity, and is that a risk or not? Could you elaborate on the consequences for clinical treatment?
Yes, so from the image about cytotoxicity, we observed that ELX02, the compound you were mentioning, did not affect in a negative way the cell's viability. And this is a very good sign. And indeed, now it's in clinical trial for Alport disease. With the clinical trial is still ongoing, so there are no results for that yet. There was results for clinical trial for cystic fibrosis. So for the safety profile, that was fine, but according to the results, it was not great in restoring the protein involved. But for safety, from our results, of course, in vitro, but also from clinical trials, it looks promising, ELX02. And so I don't think there are big risks, at least for the results that are available now. PDC, it is actually true, it's approved in Europe from European Medicine Agency for Duchenne muscular dystrophy.
Thank you very much for elaborating, but how do you explain the cytotoxicity in the in vivo experiments? Because being a clinician, I mean, I do feel kind of like, is this the way to go?
For one specific molecule in particular?
You saw cytotoxicity with all agents.
With all agents?
Yeah.
Yeah, so our experiments about cytotoxicity were performed in vitro with 3 different cell lines. So of course we do not have with this a complete overview in an organism or as an entire system of how this could affect like other organs. So I think for this compound, well, there are also other researchers that use these compounds.
Yeah.
And yeah, actually for us, for instance, diaminopurin was very toxic, but this was also tried in mice. And
Thank you very
see.
much.
So,
it's not the right model, right? That's why you can't compare it to the human situation.
Yeah, and animal model would be better for sure for cytotoxicity.
Thank you very much. I'm giving the pro-rector is...
Okay, thank you for your participation. The opposition will be continued by Professor Bernardi, Professor of Biochemistry, University of Ferrara. Thank you for coming all the way from Italy.
Thank you for inviting me. Thank you for giving me the opportunity to raise question. Dear Promovenda, it's really nice to meet you. In the future, you have really reached impressive achievement, and really, I'm really pleased about this, about your growth, scientific growth, I mean. My question focuses on the chapter 2, on the von Willebrand factor side, and also on the proposition 2. I mean the importance of the variants that we have found, both in terms of factor VIII binding and maturation of von Willebrand factor. And you have performed really impressive screening, starting from the DNA level with candidate variants, DNA level, which substantially excludes the presence of one allele in terms of mRNA, and this is very nice because at this point you have an apparently homozygous patient, which is a dream for all people who work on genetics. And at this point, you can start, of course, expressing this variant, single or in combination, and you discover that in combination, it's better in terms of expression than only alone, and that's really a surprise. And after this, you perform binding assay, for instance, for solid phase factor 8, and you discover that there is virtual absence of binding in your assay. Moreover, in simulation, you find that at the same time, you find that the binding site for furin is virtually destroyed, the sub-pocket that it binds. And this, again, indicates that the second part of the story, cleavage, is also severely impaired, okay? And when you perform expression experiment, this works. You find that your recombinant has, would say, vast majority of pre-von Willebrand and virtually traces of mature von Willebrand. But in vivo, this is not the case. In vivo, you observe that, of course, the prevailing part, protein part, is the mature form. And concerning factor 8, you find that the patient has really, would say, 40% factor 8 activity, which would not be compatible with the absence of binding. My question is, taking into account all these really nice approaches that enable you to obtain information that you have to combine, do you believe that the assay you have used, carefully used, I would say, permit you to speculate about the blood phenotype? I don't say the clinical phenotype because in this case you have additional platelet effect and so on that I'm not going to discuss. But in terms of the parallel between in vitro observation, including simulation, modeling of, to the blood side or to the plasma side, what's your impression? I think this assay gives you the right information. Are there discrepancies?
Highly esteemed opponent, thank you for your question and for your nice words about my thesis and my work. Indeed, when we performed the Western blot on plasma, we observed that two forms of von Willebrand factor were present, indeed, which was not the case from the Western blot in vitro. And indeed, in vitro and plasma are two different systems. And also in vitro, we expressed von Willebrand factor from COS cells, which are kidney cells, while in vivo, it comes from endothelial cells. So it's different, of course. So, of course, in vivo, the expression is in the natural side. And these results do not really match. But it could be that in vivo, maybe the pro-von Willebrand, so the form that is secreted with also the propeptide attached, because it's not the regular one, it could be that is actually cleared or under attack of proteases. So what we see could be also an underestimation of that. And this would rebalance with the mature von Willebrand. And of course, probably in vivo, the furin is in the natural source of von Willebrand. So it might work differently in vivo than in the COS cells we used.
And concerning the factor 8 level story?
The factor 8 level story. So the patient had 44%. Yes, which is slightly low. But with the impairment, you would expect lower factor 8, probably. Well, we find
solid binding, solid assay.
Yeah, the solid assays, of course, yeah, it's pure proteins, so you don't have the plasma context and yeah, the in vivo model. Yeah, but it does match that there is impairment of binding, and it does match with the so the new novel mutation. It actually impairs the binding. So from that point of view, I think it's a reliable model. For the percentage of factor 8, and it doesn't really as a model.
Thank you for addressing these not easy questions. Have I still 1 minute, or
Very short question.
No, it's not.
Because when you have
it's
a
not
long
a short question. So
when you have a long question,
I will
when you have a long question, you get a long answer, as you noticed. Okay, thanks for your contribution.
Thank you.
The opposition will be continued by Professor Reitsma, Emeritus Professor of Experimental Molecular Medicine, Leiden University. Also for you, thank you for coming to Maastricht.
Thank you very much, Mr. Pro-Rector. Madam Candidate, first of all, compliments on the thesis and also your promotion team obviously included. And it's, yeah, 3 of my favorite subjects I think are in here, factor 5, blood coagulation, and genetics. So what more do you want? So I'm really happy to be here and being able to talk to you about some aspects of your thesis. And I would like to discuss chapter 3 and try to go a little bit into detail, because doing the analysis that you are presenting in this chapter, you have to make many choices along the way. And I'd like to understand some of the choices that you made. And for that, I would like to go to page 56. And if we go to the second paragraph, describes a crucial experiment, I think, and that's an RNA analysis on whole blood. Factor 5 synthesized in the liver, but you do an RNA analysis on the whole blood. So where does this RNA come from, you think?
Highly esteemed opponent, thank you very much for your kind words and your question. Correct, factor 5 indeed is produced by liver and then released in circulation. It would be nicer to have, of course, liver to extract factor 5, but it's not really doable in patients. But we are lucky that we can find factor 5 also in white blood cells. So this comes from mostly from white blood cells, yeah, from peripheral blood.
From the end have a very significant level of factor 5 mRNA, and nobody knows why. And that was already reported in the early 1990s, I think, and has also been kind of used for a factor 5 Leiden assay, because you can do it really easy if you want to. But now, so you have this RNA in these lymphocytes, and that's what we are looking at if we look at figure 1C. Yeah, that's where you try qPCR, complicated stuff, and you look at the level of the factor 5. And for exon 5, 6, and for exon 18, 20, and your mutation is somewhere in the middle, exon 13. And what you see there is that the level in the patient is lower than in the control or in the parents of the patient, right? But it's still, if you look for exons 18 to 20, it's still like 35, 40%. And your introduction to the chapter has a long story about nonsense-mediated decay and what have you, but still it's 40%. Didn't that surprise you?
Yes, with the nonsense-mediated decay you would expect less. But factor 5 is particular, so it's especially exon 13. It's a very long exon, and from literature we know that longer if the exon is very long, it's less subject to nonsense-mediated decay, and exon 13 it's 2 kilobases.
Yeah, it's really
more or
long.
less, it's
Yeah.
really long. So for this, you can have less nonsense mediated decay, and on top of that, there is also a natural read-through occurring. So sometimes this premature stop codon is skipped. So there is more messenger RNA present for that. So there are these these important mechanisms that contribute, I think, to the presence of messenger RNA of factor V in this patient too.
Yeah, and to get added certainty, did you also try to see whether the stop codon is in there, actual, or that there was perhaps alternative splicing going on, because in your other chapters you also see surprising alternative splice forms, so could that also play a role, and is that covered in these experiments, or is that still a little bit open, that could also have played a role? Because you don't look at exon 13 itself, you do before, after, but not where the actual mutation is. And that's not, is that possible still? Is that still open, or is that?
Yeah, this mutation could actually provoke an alternative splicing. I'm actually checking from the sequence, so from C to T, A, G. Usually the most used alternative splicing, if a mutation creates a cryptic splicing site, usually donor and acceptor are GT and AG. Here is not that case. It could be, but it could be. I don't think so, though. Honestly, I don't think so in this case.
But maybe that's still something to check in the future.
Yeah,
if
could.
There is another
for sure.
Italian candidate coming from the lab of Professor Bernardi,
could investigate
looking for
this.
a wonderful project.
It's something
he should go
consider.
To, to Maastricht. Now do I still have half a minute, a little time? Yeah. So I also would like to go to figure 2, where you look at the thrombin generations, and I'm particularly interested in you see a positive signal in the patient when you use platelet-rich plasma. So if there are platelets in there, and supposedly factor V taken up by platelet human, and you also add collagen to the assay in order to trickle these platelets. But is it not also possible that this factor V comes from the lymphocytes? Because I assume these are frozen samples, right? There will be lymphocytes in platelet-rich plasma. It's not only platelets, but there are also white blood cells in there. You freeze it, they will explode, I think, if you freeze white cells in plasma. I think that's a certain way of killing them. So could it not be coming from the lymphocytes rather than the platelets?
Instead of platelets. So for the thrombin generation PRP, we use collagen to activate platelets. So platelets, when they are activated, they release factor V, and we believe that this signal is because of this, so activated platelets.
But did you check by not adding the collagen?
Did you check? I'm sorry, I didn't listen.
Did you check by not adding collagen? Would it also work? Do you need the collagen?
Yes, to activate the platelets.
Yeah?
Yes,
you had to
collagen.
do that. If you left it out, it didn't work.
No, we also tried without collagen, and we didn't see signal.
Okay, that's a good one.
We did not see signal. Yeah, I just had to think a little bit. Yes.
Yeah. I really like that answer. So, and I'm looking at the clock. I can see the clock. With that, I would like to give the word back to the pro-rector.
Thank you for your contribution. The opposition will be continued by Dr. Spronk, Associate Professor of Biochemistry, Maastricht University.
And thank you, Mr. Pro-Rector. Dear candidate, also my congratulations for this nice thesis, this nice work. also for your promotion team. I didn't prepare any words in the Italian language. I'm only at day 322 in Duolingo. So I could order a Zaino Nero, but I don't know what to do with a black backpack. And it may cost €10, so other than €10 I cannot pay. Given the time, and since your paranymphs didn't do anything other than standing there really beautiful or sitting there beautifully, I would like to ask one of the paranymphs to read Proposition 6.
Genetics play a role to a greater or lesser extent in all diseases.
Thank you very much. You forgot who wrote it. I guess Michael Jackson, or the M stands for something else. But the point I want to make, is this really true? If you consider other diseases like bone fractures, if you consider diseases like infectious disease, is there a genetic component in bumping into a bone fracture or bumping into an infectious disease?
Esteemed opponent, thank you for your kind words and for your question. I chose to put this proposition here because I truly believe so that genetics always plays a role in disease. Of course, if you bump into something and you broke your bone, yeah, it's not because of genetics, but maybe you have a fast healing process because of genetics. So maybe you heal better and faster than others. So on a positive way.
Yeah, and we could have a lengthy discussion on the definition on disease versus healing, because healing is second to disease stage. So with that, the genetics would not play a role in that certain types of disease.
Mm-hmm. I think genetics plays a role more than we think. So I think there are many predispositions, genetic predispositions for several diseases that we don't know about yet. Okay. I truly believe this.
Yeah, I thank you for that. Given the time, I would like to continue on another topic. And you mentioned it yourself in your beautiful introduction lecture, and mentioned that, and that's, I guess, a little bit biased from the team you are coming from, the importance of TFPI in coagulation. But for this thesis, it actually might be true that TFPI plays a more important role than the attention you gave it in your thesis. You started an elaborative search to start with morpholinos and siRNAs to recover from factor V mutations, factor V short. Why not simply use an antibody for TFPI? There are, at this moment, 4 companies producing TFPI antibodies. You could have used one of those. You mentioned also the siRNA for AT, antithrombin. That's now in phase 3. It's not approved yet. They are filing it at FDA. So why not use simply that to recapitulate the factor V short and do this elaborative work on finding alternative ways, which are most likely take a longer time before they are on the market?
We wanted to try an alternative strategy instead of down-regulating directly TFPI. It's true, we could have also aimed for that, but we prefer to go to a not direct way, so to modulate factor V short first. The modulation of the carrier of TFPI could then reflect a downregulation of TFPI itself. So we tried a different strategy.
But why? Since there are already products available, why then invest a lot of time, energy, resources, money in something new? Is that just to try it, or did you also try the antibodies?
We did not try the antibodies against TFPI. We focused on the modulation of factor V.
But what was the reason behind that? Did your promoter not believe anymore in TFPI, or?
But no, I don't think this was the reason. We really like Factor 5, and because we wanted to modulate this plus.
Thank you very much. I give the word back to the prorector.
Thank you. Alice Todaro, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Alice Todaro, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Hacking is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Dr. Todaro, are you ready for the oath?
I'm ready.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Alice Todaro, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of our university.
Dr. Todaro, dear Alice, after a long and eventful PhD journey and today's successful thesis defense, which was really impressive, here you stand at last with the PhD diploma in your hands and a proud feeling in your heart. Having shared with you all the ups and downs of this journey, I'm happy to be the first to congratulate you and your family on this milestone achievement. Complimenti. Complimenti. Your PhD was quite special in many respects, starting from the unusual circumstances that brought you to Maastricht in the first place. Maybe you remember, on a spring day in 2018, I was just about to board a plane to Italy when I received a phone call from Professor Hacking, urgently requesting my help in recruiting a PhD candidate for an exchange program between Karim and Padua University. Deadline, end of the week. Bewildered by what looked like a mission impossible, I contacted Professor Bernardi, my previous PhD supervisor at Ferrara University, who kindly offered to chase up some of his recent master graduates. 2 days later, he forwarded us your CV with a warm recommendation and a postscriptum: she might need some encouragement to apply. So we got in touch, first by mail, then by phone, and finally in person for a rather unconventional job interview over dinner in a pizzeria. And the rest is history. You arrived in Maastricht with your parents in a fully packed car at the beginning of August. Your father wanted to know every administrative detail. And when I indicated that he was not expected to join you for the employment conversation with the HR officer, he made me promise that I would do so on his behalf. At work, you quickly integrated into the lively social life and gossip hubs of our department, where Italian expats were by far the main ethnic minority. Based on your previous experience in Professor Bernardi's lab and your outspoken preference for cell-based work and translational research as opposed to quantitative biochemistry, we started with the Factor V modulation project, where you proved yourself a dedicated, reliable, and organized lab worker with a good hand, a keen sense of observation, and meticulous lab journal records. This project, which came with several unforeseen technical difficulties, was also a resilience and motivation test, although you did not seem to be particularly bothered by experimental setbacks. As long as your beloved cells grew and thrived, something on which you updated me every single morning, you did not mind repeating an experiment yet again or performing an additional control. Your efforts and perseverance were eventually rewarded with an award-winning abstract at the ESDH Congress in Glasgow and a first publication. After the COVID-19 lockdown, we decided to broaden your horizon with some patient-based research, and the available von Willebrand factor and Factor V deficiency project looked like great matches to your expertise. Particularly, the read-through mediated correction of the Factor V nonsense mutation became your trademark project, which you developed from an early stage all the way into a publishable message. For the megakaryocytes experiments, you spent 2 months in Professor Simeoni's lab in Padua, closely collaborating with the local team. And I'm very happy that Dr. Claudia Radu and Dr. Luca Spiezia, and also Dr. Spiezia Jr., are here with us today. This work was generally well received and earned you several presentation prizes at national and international congresses, as well as a first author publication in JTH. Among all aspects of our job, you thoroughly enjoyed traveling to new places to attend conferences and getting to know the famous scientists of our field. In contrast, reading the literature and writing were by far your least preferred activities, which became especially apparent when it came to writing your thesis. It took you a couple of months just to contemplate the task, and high doses of compensatory social activities throughout the following months to complete the first draft. Yet, you seemed to handle the stress much better than myself. And even towards the end, when personal circumstances put an additional strain on you, you never lost your smile and your good mood. Another unique feature of your PhD was the active involvement of your parents, who from the very beginning entertained a personal contact with me as your supervisor. Via WhatsApp, they sent cordial wishes for Christmas and Easter, comments on social and political developments in Italy, but also detailed assistance instructions for me when you were ill. So today I would like to address them as well. Gentili signori Todaro, anche a nome del professor Hacken, desidero esprimervi vivissime congratulazioni per questo sospirato titolo di dottorato di Alice. Dopo un percorso lungo e faticoso e le difficoltà anche maggiori che avete attraversato, credo che sia un'enorme soddisfazione essere qui oggi a celebrare tutti insieme questo traguardo. Sono veramente felice che abbiate potuto partecipare di persona a questa cerimonia e rinnovo di tutto cuore i miei auguri a voi e ad Alice.
Grazie Elisabetta.
Dear Alice, it's almost time to say goodbye. I wish you good luck for the new adventure on the other side of the ocean, and all the best for your personal and professional future. Finally, also on behalf of Tilman, I would like to heartily thank our scientific collaborators for their support to Alice's project, and all the committee members and Italian guests for being here today and contributing to this very special day. Thank you all very much.
Thank you for this very nice laudatio personalized. Dr. Todaro, also on behalf of Maastricht University, I may congratulate you. Wish you a wonderful job. You did an excellent presentation, although a little bit of nervous. I think you did an excellent job. You finished it nicely on time. And I congratulate you and your family, and I wish you all the best in your future career at the other side of the ocean. And with that, I close this academic ceremony. Go to the stairs for pictures.